All right. Good morning. I imagine it's morning. Maybe it's not morning for you, but um, I just want to welcome you to this presentation, this lecture on epigenetics. And I'm feeling a little bit guilty that I gave you guys this pedigree, this puzzling pedigree of the starbelly sneetches. And if you remember, I asked you to go ahead and try to figure out last time what's the mode of inheritance of the star color in these starbelly sneetches, and keeping in mind that the unshaded or the white symbols in the pedigree chart are normal, and there are two kinds of, we could call them abnormalities in the starbelly sneetches. Some have blue stars on theirs, and some have red stars on theirs. And so your task was try to figure out, gosh, what's going on here? How is this working? How's the genetics working here? You've got two different kinds of star colors showing up. It's kind of puzzling, and I wanted you to really struggle with it. I wanted you to think about um, the different modes of inheritance that you've learned about, like autosomal recessive, autosomal dominant, X-linked recessive, X-linked dominant, um, mitochondrial inheritance, all these different modes of inheritance, and apply what you've already learned to see if you could solve this problem. But as I was saying, I feel a little bit guilty because there's no way you could have figured this out because you really don't have enough information yet. But I really wanted you to struggle, and I think there's a value in struggling because actually when we struggle at a problem, we actually build new connections between neurons in our brains. And so the struggle is important. I know you may not be buying that, but it's good that you struggled. So the question is, why was it so hard to figure it out? Why was it so confusing trying to figure out the mode of inheritance of star color and the star belly sneetches? And before we go any further, and we're going to look at the answer to that, we'll get there eventually. But before we go any further, I'd like for you to go ahead and take notes on this lecture the old-fashioned way. And you might remember that what I mean by the old-fashioned way is I'm not giving you a printed study guide that you'll fill out as we go. Instead, I'd like for you on notebook paper or on your Surface or however you want to do it, go ahead and take notes on this lecture as you would in a college class where maybe you're not given a handout. And so I think that's going to be the best way to do this. The reason it was so confusing is because this particular trait, star color in the sneetches, is controlled by another genetic system that works in addition to the direct translation of certain genes. In other words, we're talking about molecules, certain kinds of small molecules that are actually placed on top of certain DNA sequences, placed on top of the genes that changes the expression of those genes. This whole field, and of all the topics in genetics, it's a relatively new field. This field of genetics, this specialty area in genetics, is called epigenetics. Epi means on top of. And so, again, we're talking about mechanisms that happen in the cells where certain molecules are actually imprinted onto or tagged onto or placed on top of certain genes, changing the expression of those genes. So let's think about a model for this. Let's think about a way that we might envision how this could work. And we'll switch just for a minute from gene sequences to written or printed sentences or phrases, like this one, for example. Eats, shoots, and leaves. Okay, so what comes to your mind when you hear the phrase eats, shoots, and leaves? Well, I'll let you think about that for a moment. Maybe you thought of something like this, a panda bear, a panda that eats shoots and leaves. You might have thought about something like that. Um, maybe. I can't think of any other creature. Well, there are other creatures that eat shoots and leaves, um, like deer, for example. I suppose rabbits, things like that. But watch what happens if we take the same phrase and put some punctuation on top of it, like a comma right there between eats and shoots, and another comma right there between shoots and leaves. Let's watch and see what happens when we put those symbols, those tags, those imprints on top of that phrase. Eats, shoots, and leaves. Maybe you thought of something like this. Billy the Kid. He walks into a saloon in the Old West and he eats a meal and then he shoots somebody and then he leaves, maybe. So you can see how, this is kind of a silly example, but you can see how punctuation placed on top of a phrase can actually change the meaning of that phrase. Now we're not just saying eats, shoots, and leaves. We're saying eats, shoots, and leaves. See how that works? We're putting punctuation on top of a phrase and it changes the meaning of that phrase. Well, this is how we can kind of think of the way epigenetics works. Epigenetics, again, literally means on top of genetics. And the specific kind of epigenetics that we're going to be looking at in this lesson is genomic imprinting. And it's really going to be the reason why we saw what we saw on the Starbelly Sneetches. That's what we're going to get to. And I know you're probably on the edge of your seat in suspense, waiting to find out what's the explanation of the Starbelly Sneetches. But trust me, we'll get there very soon. The idea or the concept behind genomic imprinting is this. The information in certain genes is only active when it passes to a child through the sperm from dad, and the information in certain other genes is only active when it passes to a child from mom through the egg. We have a very small number of genes, then, that are stamped with a type of memory of which parent they actually came from. There's a small number of genes that if they come from dad, they behave differently in us than if they came from mom. Almost like they're stamped with some kind of a memory. Now, it's not a real memory. The genes don't think. It's actually molecules that are placed on top of certain genes before dad sends them on through the sperm, and certain kinds of molecules that are placed on top of certain genes before mom passes them on through the egg. So let's look at how this works. First of all, just a little reminder, most of our gene pairs work in what's called a biallelic fashion. Bi meaning two, like my bicycle has two wheels. Bi means two, and this is the way most of our gene pairs function, in a biallelic fashion. So for example, let's say we've got a pair of genes, a pair of alleles, big A, and each allele, what does each allele do? It produces a particular protein. This is how, up until now, this school year, we've been talking about our genes. This is the way most of our gene pairs work. Now, there's a very, very small percentage of our genes that actually work in a monoallelic fashion, where only one member of the gene pair is expressed. And those gene pairs that do work like this in a monoallelic fashion, that's the way they're supposed to work. But it's a very, very small percentage of our genes. Only about 1% of our gene pairs actually work this way. And this is what it looks like. So here's a a pair of genes, a gene pair that's supposed to work in a monoallelic fashion. And what happens is one member of the gene pair is imprinted. In other words, certain tags, certain molecules, certain kinds of genetic punctuation are put on top of that gene, in effect, silencing that gene like this. So that gene then ends up making no protein. So this is what the function in a diagrammatic form, this is what the function of a monoallelic gene pair would look like. So imprinting. Imprinting here is what explains what we saw in the starbelly sneetches. But again, we'll get to the starbelly sneetches. And remember, starbelly sneetches are just make-believe, if I had to tell you that. Let's look at a real-life human example, though, that shows the results of imprinting. There is a condition in humans, a genetic disorder, called Prader-Willi syndrome, and the phenotype is quite interesting. The people who have Prader-Willi syndrome end up with severe obesity that gets worse and worse as they get older and older. Because what happens with this condition is the small part of the brain that's supposed to tell us when we're full after a big meal, let's say, isn't functioning right in these people. And so they'll finish a big Thanksgiving dinner, let's say, and at the end of it, when everybody else feels stuffed, they'll still feel like they're starving, hungry, just want to keep eating, and they can't control it. There's absolutely no way they can control this. And as a result, they end up, as they get older and older, getting more and more obese, which can lead, obviously, as you can imagine, to other things like heart disease and diabetes and those other kinds of secondary problems that result from this. Intelligence level can be very normal in these individuals, but it's called Prader-Willi syndrome. There's another genetic disorder I'd like to introduce you to that's called Angelman syndrome. And in this condition, this genetic disorder, very, very different phenotype. Children with Angelman syndrome, they're not obese, obviously. They're a lot of times somewhat thin. They have severe mental disabilities and the inability to control their voluntary skeletal muscles. And so a lot of times these kids they'll kind of walk around in a jerky kind of a pattern, different jerky kind of movements with their arms sort of suspended up above their heads, kind of moving uncontrollably, almost looking like marionettes on strings. And so for a long time, this was actually in the medical profession referred to as happy puppet syndrome. These children always are happy and smiling and even laughing a lot, just spontaneously. And so, like I said, for a long time, it was called happy puppet syndrome. That term is slowly disappearing from the medical literature because it's really not all that PC. Both Prader-Willi and Angelman syndromes are caused by the exact same chromosome accident. They're both caused by a deletion of the very same segment of chromosome number 15. And I remember for years and years, like when I was in college, I remember learning about Prader-Willi syndrome and Angelman syndrome. And at that point, this is all we knew. That sometimes when a baby was born with a particular deletion of a particular segment of chromosome number 15 on one of their two chromosome 15s, they had Prader-Willi syndrome. And sometimes kids who had this very same deletion of the very same segment of chromosome 15 on one of their chromosome 15s ended up with Angelman syndrome. And it was a real mystery as to, why is it that sometimes they show Prader-Willi syndrome and other times other kids with the very same chromosome disorder show Angelman syndrome. And it was a real mystery, an interesting mystery. This is the segment right here of chromosome 15 that's missing. A small piece between 15q11 on the chromosome and 15q13. So it's a deletion of a small piece on the long arm, within the long arm of chromosome number 15. So this is a question that has bugged us for a long time. How can the same deletion produce two very different syndromes? We'll get to that, but we've got to cover a little background information here first. About 200 genes in us are imprinted. Now we've got 20,000 genes, so we're talking about a very small fraction, like 1% of our genes are actually imprinted. Roughly half of those are active, that means expressed on the maternally inherited chromosomes, and roughly half of those are expressed or active on the paternally inherited chromosomes, the ones that came from dad. So that means the product, this product right here, this embryo, this product of a sperm and egg has 1% of its gene pairs working in a monoallelic form like this. Okay, here's how we illustrated it earlier. And these particular gene pairs, these 1% of our gene pairs, must be monoallelic in order for normal embryo development to take place. So just what is it that makes this allele inactive in this particular example right here of a monoallelic gene pair? And here's the answer. It's the adding on of methyl groups onto that gene. Methyl groups, small molecules called methyl groups placed on top of that gene, almost like punctuation on top of a message. So what's a methyl group? This is it right here. All a methyl group is a carbon atom, as you can see, with 3 hydrogen atoms attached to it. Or if you don't want to draw it like that, you can abbreviate it like this, CH3. That's how you can express a methyl group. So it's interesting to me how the placement of these molecules on top of the gene can actually silence that gene, change the expression of that gene. So these methyl groups you see are, in a sense, the punctuation that's put on top of the message. So here's the situation. So it's chromosome pair 15 that's affected in a child who has Prader-Willi syndrome, and it's the exact same chromosome disorder that causes Angelman syndrome in other kids. So here it is. Here's a diagram of chromosome pair number 15, and I'm going to personalize this a little bit. Let's just assume here that this is my chromosome pair number 15. The blue one on the left is the one that I got from Dad. The pink one on the right is the chromosome 15 that I received from my mom. Now, the other thing I want you to notice here is on chromosome 15, I have a gene pair, SS. And then I have another gene pair on chromosome 15, UU. And notice that on chromosome number 15, the S gene and the U gene are very close to each other on the chromosome. And that's the way it is in all people. But this is what my particular chromosome pair 15 looks like. Now notice S and U are two different imprinted genes. It says here on the screen the paternal allele of the S gene is active, while the maternal allele of the U gene is active. In other words, before I received that blue chromosome 15 from my dad put an imprint, put epigenetic tags, in other words, put methyl groups on that U chromosome before he gave me that chromosome number 15. And at the same time, before mom gave me her chromosome number 15, she put epigenetic tags on the S chromosome, not the U. So here's what I'm talking about when I say that about half of the genes that are imprinted in mom are not imprinted in dad, and vice versa. So mom imprints about half of those 1% of our gene pairs that we have that are imprinted. Dad imprints the other half. And I want you to look at this diagram just for a second and kind of let that sink in. So remember, before mom passes on a chromosome 15, colored pink here, she's going to imprint that S chromosome, in effect silencing that S. Dad, before he hands on or passes on his blue chromosome 15, he's going to imprint the U allele. And so what the child, you see, ends up with is a monoallelic gene pair SS, where only one S allele is working in that gene pair, the one that came from dad. The child's also receiving a monoallelic UU gene pair, where the U was imprinted in dad. Only one U allele is working in that child. The U that came from mom is the only one that's working here. So look at this diagram carefully. This is a very key diagram for us to look at and make sure we understand this completely. Now, here's the thing that's real important that we need to remember at this point. Take a look at dad's chromosome 15. Okay, that's the chromosome that I received from my dad, that blue one. Before dad passed that chromosome on to me, that might have been the chromosome 15 that he received from his mother years earlier. But get this, and this is real important. When he received that chromosome from his mother, my grandmother, it would have been pink with the S allele imprinted. You got that? It would have been pink with the S allele imprinted. But dad took that chromosome 15 and something interesting happened in the testes of my dad. I hope he doesn't mind me talking about him like this. In the testes, in effect, what happens is before dad gave me that chromosome 15 that he got from his mother, before he passed it on to me, in a sense, you can think of it this way. He stripped off the pink paint, stripped off the imprint on the S allele, covered that chromosome with his own blue paint, and put his own male imprints on the U allele. And then he passed it on to me. Likewise, look at mom's chromosome 15, or the pink chromosome 15 that I got from my mom. Now, I guarantee you, my wife and I are not going to have any more kids for several reasons, okay, that we won't get into right here. But let's just say we did have another child. And let's say just by chance, remember it's 50-50, that I could pass on to my child that pink chromosome 15 that I got from my mom. Before I pass it on to my child, what am I going to do? I'm going to strip off the pink paint, strip the imprint off the S allele, put my own male blue paint on that chromosome, put my own male imprints on the U allele, and then pass it on. And that's a real important concept we have to remember when we're talking about imprinting. Now let's just look at a problem that could occur and indeed does occur in some people. What if a child received a chromosome pair 15 like this, where this region of chromosome 15 on the blue chromosome was deleted like this? A mistake, a broken chromosome 15 from dad. Okay, in terms of alleles, what's wrong with this embryo that has this pair of chromosomes now? What's wrong with this embryo? What's wrong with this child in terms of alleles? And I'm going to let you think about that for a minute, or not a minute, just a little moment here. And then we'll come back and we'll talk about this. I hope you were thinking that in terms of alleles, here's what's wrong with this particular embryo. This embryo has no functioning S allele, and this results in what we call Prader-Willi syndrome. Okay, look carefully at the alleles here. This embryo has a functioning U allele. It's on mom's chromosome, or the chromosome 15 that came from mom, and that's normal in terms of the UU gene pair, because UU is an example of one of those monoallelic gene pairs where there's only supposed to be one functioning U allele. There's supposed to be one functioning S allele, right? But in this case here, this embryo has no functioning S allele. There's supposed to be one functioning S allele in that particular monoallelic gene pair. But this embryo has no functioning S allele, and the result here is Prader-Willi syndrome. On the other hand, if that very same region of the other chromosome 15 is deleted, the chromosome 15 that came from mom, if it's deleted, then in terms of alleles, what's wrong with this embryo right here? And I'm going to pause and let you think about that and try to answer that. And then we'll come back and compare our notes. Okay, if you said this particular embryo has no functioning U allele, then you were right. And if an embryo has no functioning U allele, the result is Angelman syndrome. So you can see it right here. The part that was deleted, and let's back up. The part that was deleted on the pink chromosome had the functioning U allele, but if that section of that chromosome 15 is deleted like this, then this embryo has no functioning U allele, and the result is Angelman syndrome. So this is how you can explain the fact that two entirely different phenotypes result from the exact same chromosome disorder, a deletion within chromosome 15. The explanation is, if the deleted chromosome came from mom, we get Angelman syndrome. If it came from dad, we get Prader-Willi syndrome. So this disorder, these two different disorders, are determined by a combination of what's going on here is imprinting, different imprinting in the two different parents, and deletion. So both of those processes account for the differences in these two different phenotypes. Now, back to the star-belly sneetches. Let's apply what we know here and see if we can now explain what's going on here with the star-belly sneetches. And I'll give you a hint. Prader-Willi syndrome is a lot in humans is a lot like the blue stars in the star belly sneetches, and Angelman syndrome in humans is a lot like the red stars in the star belly sneetches. But I think I want to keep you in even more suspense, and we're going to save the explanation here for what's going on in the sneetches until next time. So until then, have a good day.